Hello, everyone. Thanks for joining today to our third neuroembryology round conducted by Professor Harvey Sarnat. Thank you so much, Dr. Sarnat, for continuing to teach us all about the fetus brain. So today will be the transitory and vestigial structure of the developing human central nervous system. Thank you so much. I appreciate it.
Thank you very much, Khurshid, and I want to start by again expressing my gratitude to Dr. Khurshid Mohammed for for allowing me to participate in this course in neuroembryology, and I'm very grateful for that opportunity. So today I want to discuss transitory and vestigial structures of the developing human nervous system and how they might. How do I get? Oh, I have to click on it. And how to show that sometimes they can a persistent transitory fetal structure that persists beyond its needed time may lead to neurological impairment and symptoms. And finally, comparing that with the brains of other species and lower vertebrates, we go back to the principle of Haeckel in 1875 that said that ontogeny recapits phylogeny. And just to re-emphasize that neuroembryology and developmental neuropathology provide a sound basis for understanding both normal and abnormal nervous system development, and they complement genetic neuroimaging and clinical data that we are all familiar with. Throughout the human body, we have structures that are important during a phase of embryonic or fetal life, but no longer exist postnatally or may survive as just vestigial remnants that have no real purpose that's evident. And some examples of the transitory structures might be the ductus arteriosus, cranial fontanelles, the gubernaculum, which is a cord of a core of smooth muscle surrounded by a cylinder of striated muscle that pulls the fetal testis in male fetuses up from down from the abdominal wall into the scrotum. And vestigial structures, those are transitory structures that disappear after they're needed. Vestigial structures would be those that persist. The umbilicus, the belly button we all have, the intestinal appendix, coccyx, and the medial canthal ligament of the eye are examples of those. And in the nervous system, we also have transitory and vestigial structures. And timing is a key element in morphogenesis of structures that we should always bear in mind as to when these things occurred. And so here's just a list of, I'm not going to read the whole thing, but I'm going to show you examples of most of these, of transitory and vestigial structures of the nervous system that can exist only for a period of time or may persist later and cause problems sometimes as vestigial structures. We'll start with the septum pellucidum and the cavum septi pellucidi. If you look at the primitive human telencephalon, shortly after it becomes a telencephalon from the cleavage of the prosencephalon with the development of the interhemispheric fissure, we can see that it can be divided in if you do a cross section, a transverse section, it can be divided into four parts. And the ventromedial part is called the septum. And this persists in some other species, not in humans, but it does in rodents and it does in simple animals like salamanders. And it is a neural structure as much as any of the others, the hippocampus, the neocortex, or the corpus striatum. It contains neurons and glial cells and has connections that project to other parts of the brain. But in humans, the septum starts to undergo progressive atrophy beginning before mid-gestation, and it leaves a pair of thin membranes that are glial in nature and no longer contain neurons that we call the septi pellucidi, and the space between them is the cavum septi pellucidi. And in the postnatal period, the septi pellucidi of the two sides fuse into a single midline vertical membrane, which is really functionless and is a vestigial remnant. And so it doesn't have a great deal of importance in the neonate or anytime postnatally, but because of it's so prominent and easily demonstrated by imaging, either prenatal or postnatal imaging, ultrasound or MRI, it becomes sort of a marker sometimes of neurological malformations and has that importance, but it doesn't even have any functional importance. And so it has required an attention that's beyond its real importance functionally. Here's an example of two infants that have an exceptionally wide cavum septi pellucidi, CPS. CSP is cavum septi pellucidi. And you can see this in transverse section here. And you can see these are the frontal horns of the lateral ventricles, cingulate gyrus above the corpus callosum, and this is the caudate nucleus and ventricle. And you can see this is very wide, but this is a child who has a metabolic encephalopathy, stillborn, and had genetically proved methylmalonic acidemia. 33 weeks gestation. This, with this fetus was stillborn. On the right, labeled B, is an 8-day-old infant, but born at 31 and a half weeks gestation, who had Lee's encephalopathy, which is a mitochondrial disorder. And you can see in a section through the frontal lobe, you can see the extent of the cavum septi pellucidi here. And here it is again in the lateral ventricles on either side. You notice that the corpus callosum is well formed, however. And this cavum septi pellucidi is excessively wide. So even with prenatal imaging, one could suspect a metabolic disease because of the widening of the cavum septi pellucidi. Here is a 19-week fetus, again, with a very wide cavum septi pellucidi. And the reason I'm showing you this slide is to show you that it is not part of the ventricular system. So here's the lateral ventricles, ventral to it, and you can see that the lateral ventricles are lined by ependema. This is the third ventricle here, actually, and extending in, to the lateral ventricle, third ventricle in the midline, and this is the foramen of Monroe, actually. And the choroid plexus is coming from the roof of the third ventricle, but the entire third ventricle and the lateral ventricles are lined by ependema. Here it is at higher magnification in D. And fetal ependema is strongly immunoreactive for protein vimentin of incompletely differentiated cells. And you can see that the ependema is strongly reactive, but there is no ependema in the cavum septi pellucidi because it is not part of the ventricular system and does not communicate with it. Here you can see an example of ultrasound. This is a mid-gestational normal infant, and here's the cavum septi pellucidi on the left, labeled A. You can see it again on B at a different level, and here you can see it in This is the cavum septi pellucidi here, that you can see it in an axial view. These white structures here are the choroid plexi, but you can see the cavum septi pellucidi. So you can see that this can be easily identified in mid-gestation or earlier, that in fetal imaging. And it also, one can demonstrate the absence of a cavum septi pellucidi. For example, in this patient who has a septo-optic pituitary dysplasia, the septum pellucidum is absent. And you can see that that you can't visualize the septum pellucidum. This child has a wide operculum here, and it becomes actually a schizencephaly as an associated defect in this disorder, which is not uncommon in septo-optic pituitary dysplasia. But there's an absent cavum septi in this case. The next remnant I would like to demonstrate is the massa intermedia between the thalami. Sometimes in the literature, this has been called the thalamic adhesion, but that's not a very good term because it implies that it might be a white matter commissure or fascicle, which it is not. It is a fusion of the medial nuclei of the thalamus in the midline, and it's usually an incidental finding in the MRI or even neuropathologically, and it doesn't occur in all brains, but it's a very common finding, and it has no real clinical significance generally because it does not obstruct the flow through the third ventricle or cause any neurological abnormality, and it's considered to be a normal anatomical variant. And it can be demonstrated prenatally, again, by imaging in the late second and during the third trimesters. It's somewhat species specific as to the size of the thalamic adhesion, because in some species, such as horses, they have an enormous massa intermedia, which almost fills the third ventricle, yet does not cause obstructive hydrocephalus. And I know this from personal experience because I used to do veterinary, large animal veterinary neuropathology for the province of Alberta for about a decade, because I have a degree in zoology as well. And so I examined a number of horse brains, and they all showed this particular anomaly. In humans, it's excessively large, often in septo-optic pituitary dysplasia, and certainly in holoprosencephaly. And so the pathogenesis of the massa intermedia is that it's a vestige of the diencephalic cleavage at 4 to 5 weeks gestation that doesn't completely cleave between the two thalami that are forming at that time in the two what will become the two telencephalic hemispheres. And here's an example in imaging of a massa intermedia. This is an 8-month-old girl, and she has a mild obstructive hydrocephalus, but probably also had aqueductal stenosis as the cause. And in sagittal view on the left, you can see that there is a large massa intermedia that looks dark in this T2 weighted image. And here you can see it in the third ventricle, wide third ventricle, in axial views and in coronal view as well. The lamina terminalis is an important midline embryonic structure that demarcates the telencephalon from the diencephalon, and it demarks the rostral extent of the third ventricle. And within the lamina terminalis, in the most dorsal portion, it forms the bridge of the corpus callosum, and also at that point is the source of the prosencephalic neural crest tissue, which will migrate as a vertical sheet in the midline and help in craniofacial development. The middle part of the lamina terminalis forms the anterior commissure, and the more ventral part forms is where the optic chiasm forms. And in even adult brains, you can see a remnant of the lamina terminalis as a thin, transparent vertical membrane that extends between the optic chiasm and the overlying hypothalamic region at the floor of the third ventricle. And this vestige can be visualized at surgery and certainly at autopsy, but it's too thin to be resolved by present neuroimaging. And these are drawings from Wilhelm His, who is the renowned Swiss neuroembryologist who first described it. And he also described the neural crest and named it around 1875. And in these drawings of his, you can see the lamina terminalis illustrated here as this in this sagittal view as this white line, and here it is again in this more mature brain. And here's the functional timing of the embryonic lamina terminalis and when the various structures form that go through it. Now, some children who have agenesis of the corpus callosum also have hypertelorism, as you see in this infant here. And that's because of dysgenesis in the dorsal part of the lamina terminalis that I told you forms the bridge of the corpus callosum and also the source of prosencephalic neural crest tissue, which is necessary to form the intercanthal ligament that holds the orbits together during growth, so that the eyes are nearer to the midline and facing forward instead of on the side of the head. And so if you see an infant with hypertelorism, and we have good charts to show exactly what the interpupillary distance should be at each age in the fetus and in the newborn and postnatally, it's enough justification to do an imaging study to show that there's an absent corpus callosum. Here's another abnormality of the corpus callosum that we sometimes see that is really a persistent vestige, and I'll explain that. This is a 10-year-old boy who has trisomy 21, Down syndrome, and you can see that the corpus callosum is excessively thick in this child, very thick. The anterior commissure is normal. I think you can see that as a little white dot here at the base of the fornix. And the pericallosal arteries are well formed. So what does a thicker than normal corpus callosum mean? Well, between 20 and 40 weeks gestation, mid-gestation and term, there's about a 50% reduction in the number of callosal axons in normal fetuses. And then between term and 2 years of age, there's an additional 25% reduction in the number of axons in the corpus callosum. And these retracted axons are really collaterals or branches of the primary axon that may have several, 3 or 4 branches each axon. So you end up with a result of fewer but more specific and more precise connections, which is more important for functional connectivity in the brain. Myelination of the corpus callosum is a postnatal event. Just for information, all commissural axons, whether they're segmental spinal commissures or any of the commissures of the brainstem or forebrain, including the corpus callosum, are all GABAergic and inhibitory in function. So what does this mean, though, in terms of the vestiges that we're talking about? An excessively thick corpus callosum indicates a maturational delay or arrest in the retraction of axonal collaterals, so that you end up with too many axons in the corpus callosum. And it looks even thicker when they become myelinated postnatally. And so in the case I showed you here, this is a genetic problem because this child has trisomy 21. Not all children with trisomy 21 show this, by the way, but some of them do. And it's also seen in a number of other genetic disorders and metabolic encephalopathies. Neural crest cells arise in the lateral margins of the neural plate before the folding of the neural plate. And these are provisional neural crest cells that are brought to the dorsal midline as the neural plate closes to form the neural tube. And as soon as the neural tube closes, these neural crest cells migrate to the periphery to form not only nerve sheaths like with Schwann cells and other neural structures such as the autonomic and dorsal root ganglia and chromaffin tissue, but also many non-neural cells such as adipocytes, fat cells, melanocytes, the Purkinje conduction system of the heart, cartilage, membranous but not endochondral bone, and connective tissues. The tissue differentiation from neural crest does not occur until after migration is complete and the cells are in their final destination in the periphery, in the body. And so that neural crest is a transitory tissue, fetal embryonic tissue really, that no longer exists even in fetal life. It normally does not persist as the embryonic neural crest tissue, but only the derivatives of it. And in this diagram, you can see the folding of the neural plate, and you can see that if you look at the neuroectoderm white here in the placode, you can see that as it folds, it brings the lateral margins of the neural placode, which contain the neural crest progenitor cells, to the dorsal midline, and then you get migration. And here is an example of neural crest tissue in this dorsal midline at the time of closure of the neural tube in a 5-week human embryo. And this is a chick embryo here. That's very nice work done by Lamina on calcium that showed in black. The neural crest cells are marked in black, and you can see that this is mesencephalic neural crest. This is the formation of the cerebral aqueduct. But as the dorsal part closes of the future midbrain, you can see that you start to see migration, and then a few hours later, you can see the migration has progressed even further, and it's going to go out and form many of the structures that neural crest forms, both neural and non-neural structures. And here's some examples of neural crest derivatives. Here we have the adrenal medulla, the chromaffin tissue, and in the lower left, we can see a dorsal root ganglion. This is all neural crest derivative. This is the epiphyseal plate of a rib in a mid-gestational fetus. You can see that membranous bone is forming in the rib, and that's from neural crest derivative, and the cartilage that precedes it is neural crest as well. And here you can see myenteric parasympathetic plexi forming in a term neonate within the intestine, sustained with acronine orange fluorochrome. And the septum and Purkinje conduction system of the heart also is of neural crest origin, cardiac neural crest it's called. The germinal matrix refers to undifferentiated neuroepithelium in the subventricular zone, and these are post-mitotic but pre-migratory cells. And you can see here that the ganglionic eminence is in one place, and you can see that around this ventricle you have a darker staining area, which is the germinal matrix. The ganglionic eminence will give rise to glutamatergic excitatory neurons that will migrate by tangential migration, not by radial migration. And this is source of those cells in the cortex, which constitute about 20% of neurons in the cortex. Neuroblast migration is complete by 16 weeks gestation, pretty much complete. And so that there still is germinal matrix that can be seen after 16 weeks, and you can even see it in a term infant. And the cells that it's forming will be glioblasts, which will form glial cells rather than neurons. Here you can see again in a 16-week fetus. Unfortunately, this is torn. The corpus callosum is here, and it should be brought together. But you can see the ganglionic eminence and then other portions of the germinal matrix. And the dark line here are the migratory cells that are going towards the cortex. Radial glial cells are unique glial cells of the subventricular zone because they really are also sort of stem cells or progenitor cells that have a multi-potential for differentiation, although the great majority of them become astrocytes, fibrillary astrocytes, mainly in the white matter, and they serve as sort of a monorail for radial neuroblast migration. And the neuroblasts migrate along on the surface of the radial glial fiber, not inside of it. After the neuroblast and glioblast migrations are completed, the radial glial fibers retract, and as I say, most become mature astrocytes, so that radial glial fibers no longer exist in postnatal brain. And here you can see them again, and you can see them in a midget. That was a 9-week fetus I showed you earlier. And here you can see them in a mid-gestational fetus, radial glial fibers that are extending centrifugally towards the cortex and go all the way to the peel surface. The tangential migration that I mentioned earlier that comes from the ganglionic eminence does not follow the radial glial fibers. And this is the migration of those. And you can see in the mature term neonate that this is a reactivity calretinin, which is a marker of, it's a calcium binding marker of GABAergic inhibitory interneurons. You can see the distribution of these within the cortex of the term neonate. There's also the subpial granular glial layer of Brun over the cerebral cortex. It's a glial layer that prevents overmigration, and it's transitory. It disappears after migration is completed. And it covers the entire neocortex by 18 weeks gestation and reaches a maximum thickness at about 22 weeks. And then it becomes thinner by migration of these cells into the cortex, where they become indistinguishable from other astrocytes. And the layer of Brun is very sparse at term, and it disappears completely in the early postnatal period in the first few weeks after birth. There are certain malformations in which the layer of Brun is deficient. The lysencephalys is one example, holoprosencephaly is another, and that facilitates the formation of the glioneuronal heterotopia in the overlying meninges, and that results from overmigration of cells that don't stop at the peel membrane. And there's also an external granular layer in the fetal cerebellar cortex, and that persists much longer. But these are not These are not glial cells. These are progenitor neurons. And here you can see in this term neonate, the cells that provide a similar function as the radial glia of the cerebral cortex, coming from Bergmann glia, specialized glial cells in the Purkinje cell layer towards the cortex. But you can see that there is an external granular layer, and all of these neurons, these progenitor neurons, are going to migrate down into the internal granular layer for the mature cerebellum. And this is a Ki-67 on the right, which is a proliferating cell nuclear antigen that identifies mitotic potential in these cells, even if mitotic spindles are not seen histologically. And you can see that the outer part of the external granular layer still has a lot of proliferating cells that are forming more and more external granular cells to migrate. The external granular layer can be seen as much as 18 months of age still in an infant, and after that, it no longer is seen. In the cerebellum also, we can see arrested development of the rhombic lip of His, the primordial cerebellum, and that's the origin of the external granular layer. And here you can see the primitive cerebellum forming in this brainstem. This is caudal. This is the spinal cord cut off here at this end. And you can see that in the floor of the fourth ventricle, you can see these ridges and grooves between ridges, which are the rhombomeres, or initially the segmentation of the neural tube that should not be seen any longer in this infant. This was a maturational arrest of cerebellar development. Cajal-Retzius neurons are the neurons that are found in the molecular zone of the cortical plate, but they precede the very first migrations, radial migrations, from the germinal matrix. And these are large, mature neurons, and they form a pre-plate plexus in the marginal zone of the telencephalon before the first wave of neuroblast migration occurs. And they occur only in the neocortex, not in the hippocampus, amygdala, or any subcortical structures. These Cajal-Retzius neurons express the gene relin, which its transcription products are important for the laminar organization of the cortex. And if abnormally clustered, they can contribute to disorganization of the cortex and an epileptogenic condition, such as in polymicrogyria. Their origin is from ganglionic eminence, again, from the germinal matrix, the same origin as the inhibitory interneurons of the cortex that we saw earlier. After mid-gestation, the Cajal-Retzius neurons become sparse with brain growth and are difficult to find in mature brains, but they do persist and they don't disappear. And here you can see in this 24-week fetus, you can see Cajal-Retzius neurons in the molecular zone of the cortex. And you can get abnormal clustering of the Cajal-Retzius cells in the cortex of polymicrogyria. You can see in this illustration here. The subplate zone is at the other end of the molecular zone. It's at the bottom of the cortical plate and is a transitory area where you have many neurons which are similar to the Cajal-Retzius cells. And this is a layer which also helps organize the architecture of the cortical plate and the arrangement of neurons. And it is seen between 7 weeks gestation and starts to regress at 24 to 34 weeks gestation, extremely variable. And the residual can be seen sometimes even at term, but it may set limits to structural plasticity by synaptic rearrangement of the cortex. The subplate also is the origin of pioneer axons to the corpus callosum and to the thalamus, the corticothalamic projections, and permanent fibers are from other areas. In the case of the corpus callosum, they come from pyramidal neurons in layer 3 of the cortex. And the position of the subplate zone is eventually gradually replaced by the development of the U fiber layer when gyration and sulcation begin, starting at about 22 weeks gestation. The Indusium griseum is a thin layer of neurons and glial cells on the dorsal surface of the corpus callosum. It's a microscopic finding, but it denotes the migratory route of the hippocampus from the dorsal-medial portion to the ventromedial position. And it's associated, this shift in position of the hippocampus is associated with the telencephalic flexure. And the telencephalic flexure, you can see in lateral view in this diagram, bends the primitive telencephalon so that the tip of the primitive telencephalon becomes the tip of the temporal lobe, not of the occipital lobe, because of the bending. But the bending also causes the hippocampus, which starts dorsally and comes out ventrally, to migrate, the so-called migrate. And this is the route of migration of the hippocampus, but it's of course superimposed on a mature ventricular system so that you can understand that a little bit better. And here we have an example of alobar holoprosencephaly, and this is a dorsal view of this brain with non-cleavage of the hemispheres, and you can see that this band is the hippocampus, which is continuous across the midline instead of being separate, but it's also a dorsal structure because in holoprosencephaly, you do not have a telencephalic flexure. And if you look at the brain of rodents like mice and rats, they also do not develop a telencephalic flexure, and the hippocampus remains a dorsal structure. The habenula is a paired epithalamic structure. It's next to the pineal in the roof of the third ventricle and part of the limbic system. Interestingly, phylogenetically, it's the first brain structure to show an asymmetry. We talk about the asymmetry of the cerebral hemispheres and functional asymmetry in terms of many functions, including language and spatial orientation. But in primitive fishes such as lampreys and other early fishes, sharks and rays, the habenula is asymmetrical. It's better formed on one side than it is on the other. And there's variable asymmetry in other types of vertebrates. And it's generally smaller in mammals than in other vertebrates. It has a commissure between the two sides. And it receives afferent axons from a number of structures, including the septum. We talked about the septum earlier, and in humans, the habenulae are symmetrical, and they're larger in early fetal life than at term, but they don't disappear. And perhaps they become smaller and less significant because associated with the atrophy of the septum that we talked about earlier. And so this is another example of a residual vestigial structure. Choroid plexus cysts are often found in imaging and are prominent intraventricular structures in the fetal brain that occupy a larger volume of the ventricle than they do postnatally or in the adult. And routine ultrasonography at mid-gestation often reveals asymptomatic small cysts within the plexi, within the choroid plexi, that seem to mysteriously disappear at more advanced stages. And the reason they disappear is because they rupture and they no longer are cysts. They're more frequent in the choroid plexi of the lateral ventricles than they are in the third or fourth ventricle in normal brains. And there's two histological forms. In one, the epithelium is on the outside, as with the villi and fronds of normal choroid plexus, and the connective tissue base lining the cysts. And in the other, the cysts seem to be inverted epithelium facing the inside. So in that case, it's producing cerebrospinal fluid, but it's being trapped inside because that particular portion of the choroid plexus has been turned inside out. Multiple choroid plexus cysts occur in trisomy 18 and trisomy 21, and also in some congenital infections such as Zika virus and cytomegalovirus, and also in some metabolic diseases, particularly molybdenum cofactor deficiency. And they have to be distinguished from cryptic hemangiomas of the choroid plexus. So here's some histological examples of choroid plexus cyst. In a 35-week gestation infant, you can see the cyst, and you can see that the epithelium is on the outside and the connective tissue base is on the inside. In this case, Blake pouch cyst is another kind of cyst of the ventricular system, and it forms in the midline as a transitory structure protruding in the initial fourth ventricle from the base of the primordial choroid plexus, and CP is choroid plexus, and it ruptures normally during normal embryogenesis, and it leaves an opening that's the foramen of Magendie, the midline foramen, which becomes the vallecula or the fourth ventricular outlet, and that occurs before 8 weeks gestation. But sometimes it can persist even into the second trimester. If it persists as a vestigial structure, it may remain asymptomatic, but sometimes it can obstruct the fourth ventricle outflow of cerebrospinal fluid, and it can cause fetal or neonatal hydrocephalus. And it's sometimes associated with hypoplasia of the cerebellar vermis, and some authors consider it to be, therefore, a variant of Dandy-Walker spectrum. I'm not sure I agree with that, but that is described in the literature. It was first described by Blake in the year 1900. Persistent trigeminal artery is a vascular anomaly, and it's a persistence of a vessel which is transitory in fetal life. The basilar artery does not start out as a single midline vessel. It starts out as a pair of vessels at the base of the brain called the paired longitudinal neural arteries, which then fuse to form the basilar artery. And at the stage of the longitudinal neural arteries and early fusion, there are a number of transitory connections between the carotid system and this primordial basilar artery, and one of them is the trigeminal artery, which is the otic artery that's posterior to it, and there's another prominent one, rostral to it. And these vessels atrophy after fusion of the paramedian longitudinal neural arteries. They are a connection between when they persist, they are a connection between the internal carotid and the vertebrobasilar system of blood flow. And you can see that because of the folding of the brain, that these two arteries are closer together in the fetus than they would be in the more mature brain. See them in longitudinal section here. Well, here is a cerebral arteriogram of a 46-year-old woman. I actually performed this arteriogram when I was a pediatric neurology resident, and you can see that the carotid artery, and here is the posterior circulation. This is the basilar artery, and here is a persistent trigeminal artery, and all of the blood for the posterior fossa is coming from the carotid system because of the persistent trigeminal artery. And so if this woman ever had an occlusion of her internal carotid, she would lose her posterior circulation as well and have a much more extensive infarct than a person who had a normal maturation of the vascular system. Another anomaly of the vascular system is a persistent hyaloid artery. This is the hyaloid artery here in a 6-week fetus with persistent hyaloid artery. The hyaloid artery appears early in the development of the eye, of the optic cup, and it extends into the vitreous surrounding the lenticular placode that will form the lens. And it's very prominent in about 5 weeks gestation, and then it recedes at about 7 weeks gestation before closure of the optic stalk fissure. And it then disappears, and it's replaced by the ophthalmic artery, which is the mature supply of blood to the retina. A persistent hyaloid artery occurs in patients with otherwise normal brains and causes progressive visual loss in childhood and adult life if it's there. And it can cause opacification of the vitreous, can cause cataracts, and even retinal detachment. It persists in the cyclopean eye, also of alovar holochrosocephaly. And it sometimes can even be demonstrated by prenatal ultrasound. And it's something that can be treated by laser surgery and by ophthalmologists if it's demonstrated, and should be done because it'll cause progressive visual loss if it's not treated. There also are some persistent recesses of the ventricular system in non-mammalian vertebrates and in part in humans as well. So this is a drawing of the ventricular system in a shark, and you can see that there's a prominent extension of the ventricle into the olfactory bulb, an extension of the ventricle into the cerebellum, and into the optic tectum, the superior pelliculi, and a posterior recess that also can is large. And in the human, an olfactory recess of the lateral ventricle occurs in the fetus. This is a 24-week gestation fetus. This is normal. And it can be enlarged in fetal hydrocephalus, as you see in these twins that both have aqueductal stenosis and fetal hydrocephalus. But this is an olfactory bulb from one of these that shows the persistence of this of the olfactory recess. Spinal central canal. The central canal is formed by closure of the neural tube. Here's a neural tube in an early fetus, and it normally the central canal will atrophy in postnatal life and becomes obliterated, leaving only some ependymal cell clusters and rosettes as a vestige. And in some malformations of posterior fossa structures, persistent spinal central canal persists in about 30% of Chiari 1 and 60% of Chiari 2 malformations, and also frequently in the Dandy-Walker syndrome. And sometimes it becomes dilated and called hydromyelia. The C1 cervical spinal nerve root does not exist in the mature human. It's prominent in lower and other phyla of vertebrates, such as amphibians and reptiles, in both the fetus and the adult, where it innervates some upper paraspinal muscles. But in the human, it's transitory at 4 to 5 weeks gestation, and it does not innervate dermatomes or myotomes or somites or any other peripheral structure. It regresses and disappears by late embryonic life, about 6 weeks, and does no longer occur. So now I'd like to talk a little bit about human tails. In late embryonic and early fetal period, the coccyx is prominent, and a tiny human tail is transitory and is soon resorbed, so that all human fetus embryos have a tail, but it's transitory. A persistent true human tail contains vertebral structures, neural elements, but not spinal cord, nerve roots, and some nerve roots of the cauda equina, as well as striated muscle. And the tail does not always emanate from the most caudal extreme of the vertebral column, however. A pseudo-tail, by contrast with the true tail, is just a skin-covered loose connective tissue without any bony neural or muscular components, and is asymptomatic. And enhanced growth of the caudal vertebral column and neural elements can be induced by the gene sonic hedgehog, which is expressed by the notochord, or by endogenous retinoic acid, which is the alcohol of vitamin A. And that's sometimes used by women, vitamin A, for dermatological conditions before they know that they're pregnant, and it can induce human tail. And true tails may be asymptomatic, and they can be excised in early infancy for cosmetic reasons, but the patient should be observed in the first 2 years after that, because there may be progressive tethering of the spinal cord that may require further neurosurgical intervention. And in some cases, nerve root entrapment or stretching may already be exhibited at birth, neonatal period, or early in infancy. Many animals have tails normally, but humans do not. And here's an example of a toddler with a true human tail. You can see here, and if you look at the lateral view of the röntgenogram, the spine, you can see the extension of this tail, and it's segmented just as the vertebrae are segmented. Here's a different neonate, and this is a pseudo-tail that contains no bony elements, no neural tissue, no muscle, just some connective tissue covered by skin. And this is asymptomatic, and the treatment would be excision and verification of what it is histologically, but that's easily treated. And there are a number of genes involved in human tail formation that might be mutated to cause human tails. So in conclusion, the neuroanatomical morphology of the human fetal brain differs from morphology at maturity, in part because there are many transitory structures that disappear or that become vestigial in adults. And neuroembryology and developmental neuropathology are important bases for understanding normal and abnormal CNS development. And it's important to recognize the transitory structures that persist beyond their time may cause neurological disabilities later in life, as we've shown. I thank you very much for your attention.
Thank you so much, Dr. Sarnath, for this enlightening lecture. Lots of structures that we see postnatally now make sense to me. And I think we tried to enable the chat, but that is disabled by the institution. You can still write your questions in the Q&A, or if you raise your hand, I can unmute you and you can speak. So meanwhile, until people, if they have questions, they write there. One of the structures that I still don't have explanation for, and I don't know if you have insight to it, is the conatal cyst. You know why the conatal cyst, where it's coming from. Those are the cysts at the level of the lateral ventricles.
Well, I think that some of them are ependymal cysts, some are could be choroid plexus cysts. And ependymal cysts, I didn't have time to discuss all of the cysts, but ependymal cysts also occur that are separate from the ependymal cysts, from the choroid plexus cysts. But I think that would probably be the origin. I would have to see examples of one of those.
I think you mentioned something also resonated with me, that the neuroblast finishes in 16 weeks. And then really the glioblast is what is the migrating from the germinal matrix that we see in premature infant, and that explains that why they have white matter injury.
Yes.
Yeah.
That in part, yes. And the neuroblast migration is more than 95% complete by 16 weeks gestation. There may be a few later ones that migrate a little bit later, but almost all of them have completed by 16 weeks. And those are all glial cells that migrate after that.
Yeah, and then the Blake pouch was really interesting because that's where you say the what was the foramen there? Foramen of
the foramen of
Vajayjay, yeah. And that's why probably the leg pouch inflates
and
become big because of that circulation of CSF.
Exactly, and it obstructs the outlet of the fourth ventricle, so that it's an obstructive hydrocephalus. Yeah, it needs to be treated surgically.
That's wonderful. I don't see any questions there. You can always post your question in our WhatsApp group or email myself or Dr. Sarnath. Thank you so very much, Dr. Sarnath. This was really wonderful.
Thank you very much.